Vitamin B12, also known as cobalamin, and folate, or vitamin B9, are water-soluble vitamins absolutely essential for DNA synthesis, red blood cell formation, and proper nervous system function. Their metabolic pathways are deeply intertwined in a process known as one-carbon metabolism, where they act as crucial coenzymes. A deficiency of either vitamin causes a characteristic megaloblastic anemia and can have serious consequences, though their clinical presentations have important neurological differences. We begin with vitamin B12, its structure, sources, and digestion. Vitamin B12 is a complex organometallic compound with a central cobalt atom. Its two active coenzyme forms within the body are methylcobalamin and adenosylcobalamin. Dietary sources of B12 are exclusively from animal products such as meat, liver, eggs, and dairy. It is not found in plants. During digestion, dietary B12, which is bound to protein, is released by stomach acid and the enzyme pepsin. Once released, it binds to R proteins, also called haptocorin, which are secreted in saliva. The absorption of B12 hinges on one critical factor. In the duodenum, pancreatic proteases degrade the R proteins, releasing B12 once more. This free B12 then binds to intrinsic factor, a glycoprotein secreted specifically by the parietal cells of the gastric mucosa. The B12 intrinsic factor complex is stable and travels down to the terminal ileum. There, it binds to specific cubome receptors on the ileal cells and is absorbed into the body via endocytosis. This intrinsic factor-dependent mechanism is the only significant pathway for B12 absorption. Its failure is the hallmark of pernicious anemia. After absorption, B12 binds to a transport protein called transcobalamin, 2 for circulation in the blood plasma. Cells throughout the body take up the transcobalamin 2 B12 complex through receptor-mediated endocytosis. Inside the cell, B12 is converted to its two active forms. Methylcobalamin is used in the cytoplasm for methionine synthesis. Adenosylcobalamin is used within the mitochondria for the methylmalonyl coenzyme, a mutase reaction. Now we turn to folate, or vitamin B9. Folate exists in multiple forms, with the basic functional unit being tetrahydrofolate. Dietary folates are primarily polyglutamated, meaning they have multiple glutamate residues attached. Rich dietary sources include leafy green vegetables, legumes, liver, and fortified grains like those containing synthetic folic acid. Absorption occurs primarily in the jejunum. Intestinal enzymes called conjugases or deconjugases remove the extra glutamate residues to form monoglutamate folates, which are then absorbed via the proton-coupled folate transporter. Following absorption, folate is activated. Within the enterocytes, it is reduced and methylated to form 5-methyl tetrahydrofolate, which is the primary circulating form of folate in the blood. A key enzyme in folate metabolism is dihydrofolate reductase. This enzyme is essential because it converts both dietary folic acid and the metabolically generated dihydrofolate back into the active tetrahydrofolate form. Dihydrofolate reductase is the direct target of drugs like methotrexate and trimethoprim, which work by inducing a functional folate deficiency within cells. The core of this topic is the intertwined biochemical pathways, collectively called one-carbon metabolism. The first critical junction is homocysteine remethylation. This is where the methyl trap hypothesis operates. The circulating 5-methyl tetrahydrofolate donates its methyl group to the amino acid homocysteine to form methionine. This vital reaction is catalyzed by the enzyme methionine synthase, which requires methylcobalamin, the B12 cofactor, to function. The consequence of B12 deficiency here is profound. If B12 is deficient, methionine synthase cannot work. 5-methyl tetrahydrofolate cannot donate its methyl group and becomes metabolically trapped. This depletes the cell of other essential folate cofactor forms, particularly those needed for DNA synthesis. The second major part of one-carbon metabolism involves thiamidylate and purine synthesis. Thiamidylate synthesis is a folate-dependent step absolutely critical for DNA production. The enzyme thymidylate synthase uses the folate cofactor 5,10-methylenate tetrahydrofolate to convert deoxyuridine monophosphate into deoxythymidine monophosphate, which is the unique thymidine nucleotide for DNA. This reaction oxidizes the folate cofactor to dihydrofolate, which must then be recycled back to tetrahydrofolate by dihydrofolate reductase. Furthermore, folate cofactors, specifically 10-form L-tetrahydrofolate, are required for two separate steps in the synthesis of purine bases, adenine and guanine. The primary consequence of deficiency for both vitamins is megaloblastic anemia. The common underlying cause is impaired DNA synthesis due to a lack of thymidine and purines. This leads to a characteristic morphology. The bone marrow shows large, immature erythroid precursors called megaloblasts. The peripheral blood smear reveals macrocytic red blood cells, reflected by a high mean corpuscular volume, often oval-shaped macrocytes, and hypersegmented neutrophils with 5 or more nuclear lobes. This anemia results from ineffective erythropoiesis, meaning the abnormal precursor cells die prematurely within the bone marrow before being released. Beyond anemia, there are distinct neurological and other manifestations. Vitamin B12 deficiency causes specific neurological damage, known as subacute, combined degeneration of the spinal cord. This involves demyelination of the dorsal and lateral columns. It presents with symmetric paresthesias, sensory ataxia, spasticity, and weakness. This damage is linked to disruption of the adenosylcobalamin-dependent methylmalonyl coenzyme, a mutase reaction, and possibly impaired myelin synthesis due to methionine deficiency. Folate deficiency primarily causes anemia. It may be associated with general neuropsychiatric symptoms like depression or cognitive decline, but it does not cause the classic spinal cord pathology seen with B12 deficiency. Importantly, deficiency of either vitamin leads to elevated levels of homocysteine in the blood, known as hyperhomocysteinemia, which is an independent risk factor for atherosclerosis and thrombosis. The causes of deficiency differ between the two vitamins. For B12 deficiency, major causes include malabsorption syndromes. The classic is pernicious anemia, an autoimmune condition destroying gastric parietal cells or intrinsic factor. Other causes are gastrectomy, terminal ileal disease like Crohn's disease, and bacterial overgrowth. Inadequate intake is seen in strict vegans and some elderly individuals. For folate deficiency, causes include inadequate dietary intake from poor diet or alcoholism, increased physiological demand during pregnancy, lactation, or in hemolytic anemias. Malabsorption from conditions like celiac disease, and drugs that inhibit dihydrofolate reductase, such as methotrexate and trimethoprim. Diagnosis relies on key laboratory findings. Initial tests are a complete blood count showing macrocytic anemia and a peripheral blood smear revealing oval macrocytes and hypersegmented neutrophils. Direct vitamin levels are measured: serum B12 and either serum folate, or, more accurately, red blood cell folate, which reflects tissue stores. Metabolic markers are crucial. Serum methylmalonic acid is elevated specifically in B12 deficiency and is highly specific. Serum homocysteine is elevated in both B12 and folate deficiency. For confirming pernicious anemia, testing for intrinsic factor antibodies is highly specific, while parietal cell antibodies are less so. Treatment principles are vitamin-specific. For B12 deficiency, the standard is parenteral administration, either intramuscular or deep subcutaneous, using hydroxycobalamin or cyanocobalamin for initial loading and then maintenance. High-dose oral B12 can be effective for simple dietary deficiency. For folate deficiency, simple oral folic acid supplementation at 1 to 5 milligrams daily is sufficient. One critical clinical rule must be emphasized. Never treat a megaloblastic anemia with folate alone before ruling out B12 deficiency. Supplementing folate can partially correct the anemia, masking the deficiency, while allowing irreversible B12-related neurological damage to progress unchecked. Thank you for watching. Don't forget to like, share, and subscribe. See you in the next one.